Good afternoon, Nishika. How are you doing? So shall we begin meiosis or any other chapter? Like what's going on in your school currently? Because we have just finished the genetics chapter in the last class. Have they started any commenced with any new chapter or the same is going on? Mm. Have they commenced any new chapter, Shika? Or shall we continue with meiosis or something else? Like whatever you want. Okay, okay. Okay, so that's a very intricate topic. Meiosis, very essential for genetics, molecular biology. It's like cumulative topic, and it has multiple myriad of, you can say, events that are, you can say, extremely important and extremely relevant towards the course of expression. Expression means formation of proteins, like which proteins are produced, which proteins are underproduced, overproduced, optimally produced in various phases of the cell cycle. Okay, so first thing is what I'll tell you is, okay, okay, fine. So we'll continue with this thing only. And how about the test, Jessica? Like you were telling me a test is gonna happen soon. Like you wanted, have you got any worksheets from the school or something, or some kind of homework, or your curriculum related worksheet that maybe available in your website that you might be following, official website, something is there? Okay. How did it go? Were you able to solve all the questions? Did it have any difficult questions that were okay, okay. So how did your test go in school? I hope you did well. Okay, good, good, Ishika. Okay, so let's begin meiosis. So first of all, let me acquaint you with the term known as cell cycle. Okay, so it's a very, you can say, frequently used term. Okay, the cell cycle means how does a cell move from one phase to another, and that is the cycle which is committed towards the ultimate division of one cell into two cells. If you are talking about mitosis, and the ploidy remains the same here. Okay, or one cell which is diploid becomes or gets to become four cells with haploid nature. Okay, so this is known as a reductional division, whereas this is known as an equational division. This is what it's all about, meiosis and mitosis. So the end product or the aim, the penultimate aim you can think of, is to divide the cell. If you don't divide the cell, many cells they divide, again many cells they don't divide. So that question will also be addressing, like the intestinal epithelial cells, the hematopoiesis means the formation of various blood cells in the red bone marrow, then hair follicles, skin cells keep on dividing, right? But again, cardiomyocytes, the heart cell, neurons, the nerve cells, right? Skeletal muscle cells, they do not divide. They have a specific myosatellite cells which serve as the stem cell. Even neurons have neuronal progenitor cells, cardiac progenitor cells. Okay. So what happens is they don't divide actually. But why don't they divide? If you talk about a neuron, they do not have centriole. You might have heard of this organelle, centriole. It is present in the centrosome. Now, centriole, if it is absent, you wouldn't be able to form microtubules. Okay, microtubules are something known as a part of cytoskeleton. Okay, which are made up of alpha beta tubulin dimer. Alpha tubulin, beta tubulin, they combine together to form microtubule, whereas where each alpha and beta are associated with a GTP, guanosine triphosphate. Okay, And the centriole or centrosome is the empty OC, microtubule organizing center. Microtubule organizing center. Meaning that from this centrosome where centrioles are present almost at right angle, and you have the pericentriolar material also present, okay, the microtubules emanate. The origination happens from here. So the microtubules, they originate from here. So this is known as an MTOC. Whereas for cilia, flagella, which are also microtubule, the basal body is the MTOC. Okay, in that case, and then it binds to the centromere, which is surrounded by proteins known as chromosomal passenger complex slash kinetochore proteins. So which bind to the centromere. These microtubules are known as kinetochore microtubules. You might have seen this diagram. Then you have polar microtubules which overlap. Then you have this astral microtubules. Okay, these are the polar ones. These are the astral ones. Now, if centrioles are absent, can you think of division? You cannot go through division because you cannot divide this two chromosomes in the end. If centrioles are absent in neurons. Neurons are also arrested in many stages of the cell cycle. Now, if we talk about prokaryotes and eukaryotes, the prokaryotes do not have any cell cycle. Directly, they'll go for mitosis because they don't have any organelles. No need to duplicate the organelle. Directly, you can go for DNA division. Then you can say cytokinesis. But eukaryotes, when we talk about, okay, specifically the eukaryotes, these are attention here. We have four phases in the cell cycle. Okay. First of all, it is the G1 phase, then S phase, then G2 phase, then M phase. Before that, we have also known as G0 phase, which is known as quiescent phase of the cell cycle. G stands for gap, gap 1, gap 2. S stands for synthesis. M stands for mitosis slash meiosis, the type of division that you are considering. Okay. Now, G1S, G2M. What happens is sometimes the cell gets arrested in the G0 stage of the cell cycle, meaning that it cannot commit to the cell cycle. It cannot commence the cell cycle whatsoever. It just cannot. Okay? It cannot just commence the cell cycle. The thing is, what happens is, suppose in case of, you might have heard of, you can say reproductive cycle, where I'll just give you a very prominent example of what is G0 cell. Okay, G0 stage. What happens is, you might have heard of, like, the females, they get the menstrual bleeding. The first bleeding happens at the age of 10 to 12. At that time, they are said to have attained something known as puberty. And the first menstrual bleeding event is known as menarche. Okay. Now, before menarche, when the female is born, okay, when the female is born, you have almost, you can say, 40,000 primary oocytes which are viable. Okay. In each primary oocyte, meaning that from where ovum will be produced, okay, it is surrounded by, suppose this is the primary oocyte, this will be surrounded by zona pellucida, okay, that is a layer, acellular layer, okay, acellular translucent or non-cellular layer, and then zona pellucida will be surrounded by a layer of follicular cells known as granulosa cells. Okay. Now what happens is, you have gap junctions here, okay, cell adhesion proteins. So you'll have the integrin, cadherin, selectins, okay. So the gap junction will be open, and what happens is the oocyte, it produces cyclic AMP, a specific molecule, cyclic adenosine monophosphate, double phosphodiester bond, okay. And also these granulosa cells slash follicular cells, they also produce CAMP and also CGMP, cyclic guanosine monophosphate, and both they are able to cross, or you can say go through the zona pellucida and reach the primary oocyte. Now what happens is this cyclic AMP, it will inhibit mitotic promoting factor, means it will keep the cell arrested in the, you can say, one of the stages of meiosis, you can say. Okay, this is also a fact that it keeps the cell arrested in meiosis, so the meiosis cannot begin. Especially I'll teach you in meiosis there is a specific phase known as, in prophase 1 you have diplotin, so it keeps it arrested. Now what happens is, because of this inhibition, the CMP will inhibit MPF, meiosis arrest will happen. And then what happens is there is a specific enzyme known as phosphodiesterase 3A. If this degrades CAMP, it will form 5' AMP. Then it will be, this inhibition will be, you can say, shut down, and again meiosis would begin. But this is inhibited by this molecule, cyclic GMP. So like this, the cells, they keep on inhibiting the cell cycle. Okay. So sometimes it is even arrested at the G0 stage. So you need to come out and break the arrest from the G0 stage. Okay, this was one of the examples of arrest. This is arrest in the diplotin stage. So like that you have arrest in G0 stage also. So if you can come out of the G0 stage, what you will enter is, you will enter the G1 stage. At G1 stage, what will happen is the organelles present inside the cells will go for duplication. Because when we think of cell division, we have to divide the organelles into, you can say, or you have to duplicate the organs also. Not divide, you have to duplicate the organs also. Make Xerox copy of the organs. Because suppose you want to make your own clone. You just cannot divide yourself into two parts. You have to make 2 hearts, 4 lungs, 4 kidneys, right? Then 412 bones, a double brain, everything double. 4 ovaries, like that. So 2 times small intestine, 2 times large intestine, 2 times stomach, 2 times, you can say, pancreas, liver, okay, spleen, gallbladder. So all the organs, in the case of cell, all the organs, mitochondria, peroxisome, ER, Golgi apparatus, must be doubled, because you need to form 2 cells from 1 cell, so they will be doubled. Then we come to the S phase. S phase means the DNA needs to be doubled or not. Otherwise, how will you divide the cell? So here DNA duplication, which is also known by the phenomenon of DNA replication, takes place. Okay, DNA replication takes place in the S phase of the cell cycle. Then we enter something known as the G2 phase of the cell cycle, that is known as gap 2. Very, very important. Okay, so gap 2, the main significance is gap 2 is little bit different. Okay, so once you talk about G2 phase, okay, of the cell cycle, so it is little bit different. So how it is little bit different? What happens is, in G2 phase, the main thing what happens is the DNA repair pathway is the most active. So even the DNA repair pathway has been awarded with the Nobel Prize also. Okay, that G2 phase will concentrate on DNA repair, because during the replication process there may be many errors that could happen. Okay, in the G2 phase, or sorry, in the S phase, like you have a DNA like this, A, T, G, C, C, G, T, A, G, C, T. Suppose this is the parental DNA strand, you want to make a daughter DNA strand 5 to 3 prime, and suppose instead of P here, C, then C, G, G, again G, suppose, then A, T, C, G, A. So what happens is, this is a mismatch, because G should be C, again T should be A, this is again a mismatch. So the polymerase, DNA polymerase enzyme, okay, DNA polymerase enzyme. In eukaryotes, you have DNA polymerase alpha, epsilon, delta, omega, zeta. Sorry, you have gamma. So many DNA polymerases are there. Now, what happens is, if you talk about DNA polymerase, they are not completely accurate. You can say fidelity is not 100%, or they are not 100% fidel in nature. Okay, that is the meaning of, you can say, committing errors. So they may commit some errors which will lead to mismatch, and then mismatch will lead to mutation, like I told you, because these will be, again, their complementary will be done later on. So you will be having, you can say, the mismatch will be converted to mutation in the next cycle, right? So what happens is, the G2 phase, okay, will be responsible for DNA repair, because there are various DNA repair mechanism enzymes, okay, which are not in your course, but I'll tell you that there are many DNA repair mechanism styles or ways. You can have base excision repair, the BER pathway, nucleotide excision repair, nucleotide excision NR pathway, okay. Then you have MUT H, MUT-L pathway also. These are not in your course, but these are basically the DNA repair mechanism enzymes like UVR A, B, C, D. These are nucleotide excision. They will completely take out that nucleotide, not only the base but the associated phosphate group and ribose sugar. Then you have DNA glycosylases. They will not take out the nucleotide. What they'll do is they'll only glycosylases. So what they'll do is they'll only take out the guanine and leave the, you can say, phosphate group and deoxyribose sugar, making them apurinic or pyrimidinic site, and then they will be changing it with C, cytosine. So this happens, and when the DNA repair pathway fails, then we get something known as cancer. We may get cancer then, okay, later on. So that is the meaning of, you can say, G2 phase. Okay. In many books also, if you see G2 phase, it is also written that the organelles of protein that develop in preparation for cell division and cell grows more in size. Okay. In S phase, one thing I'll tell you, very, very important. In the S phase, what happens is, along with DNA, the centrioles also double. This is a 1 mark question, very important. The centrioles that develop, it's an organelle, but they don't develop in G1, they develop in the S phase. Okay, and then M phase will lead to directly the division of the cell. Okay, so this is the overview of the cell cycle. Okay, this is the overview of the cell cycle. Now, is it clear, Ishika, the overview of the cell cycle? G1 phase, G2 phase, S phase, and M phase, along with G0. This is the overview of the cell cycle. What happens, what all phases are there. Then we will deal each phase in detail. Okay. So any queries here? Okay, good. Okay. So now coming on to what is the real mechanism behind shifting from one phase to the other, from G1 to S, from S to G2, from G2 to M. What is the real mechanism? So what happens is, the real mechanism is, I'll tell you what is the real mechanism. What we have is, in G1, there are two proteins. Okay, in every step of the cell cycle, there are two proteins. Always remember. One is cyclin, one is CDK, known as cyclin dependent kinases. Okay. Let's talk about two proteins individually. Okay. Now, cyclin, the level of cyclin, it fluctuates. Very, very important and fundamental thing. The level fluctuates. The level of the specific CDK, cyclin dependent kinase, okay, these are the enzymes, their level does not fluctuate. Almost it remains constant. Okay. Their level does not undergo fluctuation. That is the first fundamental difference. Now coming on to, you can say, kinases. What are kinases? Okay, kinases are a type of enzymes. Okay, very very important. I'll tell you. Kinases are a type of enzymes. First of all, they are enzymes. What they do is they will cause phosphorylation. Okay, they will cause phosphorylation. Now, why is phosphorylation important? Now you will be asking. It is a very good question in itself. Now understand biology from the root. Just know for a fact that without phosphorylation and dephosphorylation, meaning that this PO4 group, PO4, O minus, O minus, this is known as PO4, 3 minus comes from the phosphoric acid present in our own body, 3H plus plus PO4, 3 minus, because our body is 70% water. So every dissociation happens in the blood, which is polar, right? Water proteins. Now, this phosphate group considered as capital P, it is added to, suppose, tyrosine amino acid of any protein. Suppose a protein has 10,000 amino acids. Amino acid 999 is tyrosine. This got phosphorylated. That is known as the residue number of protein. Okay, so 999th residue of the protein, which is tyrosine, it got phosphorylated. Or there are amino acids like serine slash threonine. They can be phosphorylated either this one and this one. Then the phosphate groups can be removed also. These covalent bonds can be broken. Okay, that is known as dephosphorylation. So many a times, in order to activate a protein, you need to phosphorylate that protein. And in order to deactivate the protein, you need to dephosphorylate. Many a times the opposite is also true. Phosphorylated protein may be inactive, then you need to dephosphorylate it to make it active. So both cases are true. But 60 to 70% of the time, phosphorylation activates, dephosphorylation inactivates. But that's not an universal law. Okay. Maybe many times phosphorylation can cause inactivation also. Okay. So get that in mind. So biology is a very, very, you can say, a dubious thing, because there is no rule which is universal here. Because it's our body, it doesn't follow a specific rule. Various exceptions are there. Okay. That's how we are the most complicated machines. And that's how we are the most intriguing machines also, which no engineer can replicate. Okay, not even remotely. Now, CDK kinases. Okay, now coming on to the kinases. So when we talk about enzymes, I think might have heard of enzymes, that they are biocatalysts. They speed up the rate of reaction, and they also are responsible for, you can say, decreasing the activation energy. Okay, so enzymes we'll talk about later on. Just think of enzymes that they speed up the rate of reaction, because that's the different chapter altogether. What is activation energy, potential energy of the entire, you can say, reaction kinetics, that I have to teach. But basically, in layman term, enzyme catalyzes, or you can say catalyzes a reaction and increases the rate of reaction. They are like the biocatalysts. The catalysts in chemistry, same work is done by the enzyme biocatalysts. Okay. Now there are 6 types of enzymes. Classes are 6. Okay. You have hydrolases, you have isomerases, you have ligases or polymerases, you have lyases, you have oxidoreductase, you have transferase. So kinase falls under the category of transferase enzymes. Okay. Now, what is transferase? The name suggests that it will transfer something. Okay. By causing some lysis. Ase means lysis. So, what does a kinase do? Okay. Kinase, it makes use of ATP. Okay. Lyses it. You have ADP plus PI, and this phosphate group, that is PO4 3 minus this PI, okay, this phosphate group is transferred to some amino acid residue, let's suppose tyrosine, of a protein. Okay, now the protein has got phosphorylated. So that is the work that kinases do. They'll phosphorylate some proteins, amino acid, at a specific residue, and then that protein will become active. The protein will be activated mostly. Sometimes it may be inactivated also, but mostly it will become activated. That is the work of kinase. Now, the problem with CDK is its kinase activity is inactive on its own. Okay, what happens is unless it can bind to cyclin protein, specific cyclin protein, what will happen is the cyclin binding to CDK will cause some conformational change in the CDK, which will, you can say, open up its active site, stabilize its active site because it is an enzyme, and will activate this enzyme, and then this enzyme can perform this kinase task or its kinase activity. Otherwise, without binding to cyclin, okay, only CDK is inactive, and thus it cannot cause phosphorylation. Okay, binding of cyclin with CDK makes CDK active and then can cause phosphorylation of the target protein. Okay, so what happens is that's why cyclin levels they keep on fluctuating, because as per the requirement cyclin levels will rise, they'll bind to their target CDK, the CDK will be active, they will phosphorylate their target proteins, and those proteins after getting activated will help in the perpetuation of the cell cycle or transfer of the cell cycle from one stage to the other stage. That is the clear-cut concept behind kinase, CDK, and cyclin. Okay. So now tell me, Shika, if you have any doubt in this specific topic. This is a very, very important, significant conceptual understanding. Any doubt here? Kinase, cyclin, CDK, any doubt? How does cyclin activate CDK? Why is CDK inactive? Okay. Why is cyclin needed in the first place? Okay. What are kinases? CDK is a type of kinase. Okay. Kinase are the transferase enzymes. So any doubt here in the technicality? Any queries? Because once you understand this technicality, the entire chapter is very, very easy. And you can just skim through the chapter with ease. Right? This is the very fundamental basic of that chapter. Okay, good. Now, I'll give you a table. Okay, depending on, just go through the table, keep it with you always. Then you can be the master of the cell cycle. Okay. So here we'll put CDK. First CDK. Then we'll put cyclin. Then we'll put the function. Then we'll put, you can say, simply I can say general name. Okay, 4 columns. Now, number 1 CDK, CDK1. There is a specific CDK, and each CDK binds with its specific cyclin. Okay, always know the rule. Specificity is the key. CDK1 can bind with either cyclin A or cyclin B, no other cyclin except cyclin A or B. Cyclin A, cyclin B, it can bind. Okay, its function is in the mitosis phase. Okay, means when the cell cycle enters the mitosis phase, the M phase, okay, of the cell cycle, then cyclin A CDK1 or cyclin B CDK1 will be active to carry on the mitosis. Otherwise, mitosis cannot begin. That is the relevance here. Okay, in mitosis, what we'll be getting is the level of cyclin A and B will be high, and the CDK1 will be activated. So it's known as mitotic CDK. General name is mitotic CDK. That is, you can say CDK1 cyclin A or this complex CDK CDK1 cyclin D. Okay, this is the complex. Next, coming to CDK2. This cyclin binds with cyclin E or cyclin A, not cyclin B. Okay, either cyclin E or A. This is important for the cell to enter the S phase. Okay, just entering the S phase. Without that, entry into the S phase. Without that, the cell cannot enter S phase. That is the phase previous to the M phase. Okay. So they will be known as what? G1 S phase cyclin or simply S phase CDK, because after G1 you have S phase. So you can call it G1S or S phase CDK, meaning that you can write CDK2 combination with either cyclin E or CDK to combination with cyclin A. Okay. Now coming to the third cyclin, okay, just elaborate the table. Keep this table with you always. Okay. Many books this will not be given. It may confuse you later on. If this table is with you, you can solve any question. Okay, from cell cycle. Now CDK4. There is no CDK3. There is CDK4. CDK4 binds with cyclin D. See, each CDK is specific for their own cyclin. That's what I was telling. Now, this is responsible for entry into the G1 cycle from G0. Okay, if at G0 you have cyclin D overexpressing and then you have CDK4, then the cell can come out of the quiescent phase of the cell cycle where there is no cell division and come to the G1 and it will initiate or commence the cell cycle event. So entry into the cell cycle G1. Okay. G1 means entry, the very first phase of the cell cycle. Entry into the cell cycle. Okay. Next, this will be known as G1 CDK. Okay. Meaning that CDK4 cyclin D. Now you have one more cyclin, sorry, CDK6. This also binds with cyclin D. Okay. Same function, G1 entry into the cell cycle. Entry into the cell cycle. Since cell cycle begins with the G1 phase, again they will be known as G1 CDKs. That is CDK 6 cyclin D. Cyclin D. So keep this table with you always. It will be of extreme help. Always remember it like this: DEAB, G1, S, then you can say SG2, SG2, and then M. So DEAB is the rule. D is G1, E is, you can say, S, E and A, and then DEA. Here it comes B. B you enter directly mitosis. So G1S, there is no specific cyclin CDK for G2. Then you enter S phase cyclins only, they are highly expressed in G2 and then you enter the M. Okay, so DEAB is the rule for cyclin, and the rule for CDK is go from the higher number to lower number, 6421. Okay, 6421. That's all. DEAB 6421. That is how it will stay in your head forever, right? So this is the trick to know this table from the depth. Okay. So any doubt in this table, Ishika? Any doubt in this table? Okay. So let's proceed again. Now I'll just show you one thing. There are also something known as, you can say, CAK and CKI. Okay, so those are also special proteins. Okay, so I'll tell you about those proteins also. So CAK protein. Okay, now CAK means what? So CAK in cell cycle, if you see, those are known as CAC protein. Okay, cyclin dependent kinase activating kinase. Okay, that's another protein that I'll just tell you the name. Okay, CAC means cyclin dependent kinase, which is the CDK activating kinase. Activating kinase. Meaning here C stands for CDK, then activating kinase. So CDK activating kinase is also known as CAS, meaning that suppose this is the CDK and cyclin has bound to it, it has got bound to it, and conformational change has happened, still this kinase is inactive. Okay, then sometimes if the kinase is still inactive, the CDK's kinase activity is still inactive. Sometimes it happens. Okay, again biology is a subject of exception, so sometimes it still remains inactive. In those cases, what happens is the CAC, it will phosphorylate a specific, suppose tyrosine, it has phosphorylated present on the kinase, and then this kinase has become active. That is the role of CAC. Okay, and then you have CKI. Okay, CDK, you can say inhibiting kinase, you can say CKI. Okay, so CKIs are also there. Okay, so CKI is cyclin dependent kinase inhibitor, you can say. So here what will happen is just opposite of this. This is CDK inhibitor. So opposite of this will be true here. So what will happen is, suppose you have CDK and cyclin. Now suppose it has become active, but the CKI will get bound to it and will make this entire cyclin CDK inactive. So this is how it can do it. Okay, so it interacts with the cyclin CDK complex to block the kinase activity. Okay, so that is how these CKIs can perform their function. More than that, you have INK4 and all. SIP, you can say family INK4, so that is not in your course, so we'll not go into the detail, but just I acquainted you with these two specific molecules. They can, you can say, further alter the state of cyclin CDK proteins. Okay. Now coming on to the next topic, just one example I'll give you of how does everything happen. Okay, like how does the cell really perpetuate or get transferred from one specific event or one specific state to the another state. Okay, so let's take the example of E2FRB. Okay, E2FRB, one example I'll give you. E2FRB complex. Now E2F is a transcription factor. Okay, meaning that it will recruit RNA polymerase. Okay, transcription factors do what? This is the gene. Suppose this is the promoter sequence upstream of the gene. The transcription factors in eukaryotes will recruit RNA polymerase, enzyme, and then mRNA could be produced via RNA polymerase. This is the layman term function. Okay, you have multiple mechanisms of that. You have type 2 RNA polymerases, type 1, type 3, okay, TF2A, TF2B, TF2H, TF2E, TF2D, so many you have. But this is the fundamental process, okay, which happens. They recruit RNA polymerase. RNA polymerase, once it binds to the promoter region upstream of the gene, it will produce mRNA, messenger RNA. Okay, and messenger RNA will bind to the RER, the ribosome, and go for the protein synthesis. Okay, so this process of producing mRNA by using DNA as a template is known as transcription. Process of protein synthesis from mRNA when it binds to the ribosome on ER is known as translation. Okay, so replication, transcription, and translation. Now E2F is a transcription factor. Okay, so during the G1 stage, okay, during the G1 stage, E2Fs are held inactive. Okay, how? Because they are associated with a protein known as retinoblastoma protein. Okay, this is the mechanism. During the G1 stage, the E2F are completely inactive. How? Because E2F are bound to retinoblastoma protein. RB means retinoblastoma. Now they are held inactive by binding to retinoblastoma E2F. Okay, E2F RB complex means here E2F is completely inactive. Okay, means it will bind to E2F and change its conformational structure, and thereby it cannot bind to its target DNA or the promoter sequence. It will be completely inactive. Just like CDK was getting activated by binding to cyclin, opposite is happening here. RB is inactivating E2A by binding to it. So both the cases can happen in biology, right? So here it is becoming inactive. Now G1 CDK, okay, just now you saw CDK4 and CDK6 are the G1 CDK. Now G1 CDK, when cyclin D level also rises, so G1 CDK meaning that CDK4 cyclin D and CDK6 cyclin D. When cyclin D level rises at the G1 stage, it will bind to the CDK4, CDK6, and then cyclin 4 and CDK4 and CDK6 are now both are active. Okay, now both are active. Now, when both are active, what happens is, what you get is, you get something known as a specific event. Okay, that is, the CDKs will do what? They are the kinases, so they will activate E2F how? By phosphorylating RB. They will cause what? These complexes, suppose this one will come and will phosphorylate. RB is a protein, so suppose on the tyrosine residue, it phosphorylated. Now, I told you that phosphorylation is always not activation. Okay, so what it will do is, this phosphorylation will inactivate the RB, and then E2F can activate the various proteins which are needed to take the cell out of the, you can say, G1 stage into the S phase. Okay, so when E2F are bound to RB, they can function as transcriptional repressor, but when E2F is bound to RB, which is phosphorylated, they will again act as transcriptional activators or transcriptional factor. Okay, so always this is the case. So this RB is then this now E2F bound to RB, which is phosphorylated by this one. Here E2F is completely active. Okay, and now it can make the cell proceed from G1 to the S phase. Okay, like that the cell can proceed from one phase to the other. So this is how you can say the perpetuation of the cell cycle keeps on happening. In the later stages, you will see ink pathway, FPGC pathway, but again those will not be talking because they are not in your course. Okay, but it's a very interesting chapter if you read in depth. Okay, so one of the things, just I told you, the for your information, that this is how it happens. Otherwise, you will keep on thinking that how does it happen? How does one go from G1 to S? Okay, just if I say you go from G1 to S, you will be kind of perplexed. You won't get the concept. So this is how you go from G1 to S, and again from S to G2, you will be having APCC proteins. Okay, different proteins will be there. Okay, so various checkpoints. So this is a kind of checkpoint, you can say. G1 to S is a kind of checkpoint, means when the organelles are completely duplicated, then only the cyclin D level will rise and the cell will cross the G1S checkpoint. Checkpoint means it's like a tool booth in the, you can say, sports races that you might have played, Need for Speed or something like that. So you might have played the tool booth races, right? You have to complete this in 10 seconds. This is specific distance. So it is just like that. So when the this event is orchestrated in such a regulated manner that you can just ponder upon the fact that when the organelles have been duplicated, then only D will the level of cyclin D will spike up, it will get elevated, and then it will activate CDK4 and 6, and then you will be able to transit from G1 to S. Before that, if it happens, before you transit, there will be a problem because the level of, you can say, the number of organelles have not divided completely and you have reached the S phase, meaning that when you reach the M phase, you may unevenly distribute the organelles, and the cell which has received the lesser number of organelles or no organelles because duplication has not taken place will be, you can say, useless cell. It will die. So that level of organization and, you can say, regulation is present when it comes to, you can say, the level of the cell cycle regulation. So this is how it is regulated. Okay, so then we will come to S phase, then G2, and then meiosis. Okay, S phase is simple DNA duplication, then G2, and then M phase. So that we'll be covering in the next class. So mostly we'll be talking about then mitosis and meiosis. Okay, so this is the concept behind or pertaining to the human cell cycle. Okay, so that you should be aware of. So you tell me, Ishika, did you have any doubt in today's lecture in the comprehension of any concept, because this was hardcore concept? Any conceptual problem? Any queries? Any clarification? Okay. Okay, good. So you have been able to grasp well. That's a very good thing coming from your side. Been a very good student till now. So, okay, then stay safe. Go to school. Okay, your school is off right now. Just I remember. Okay then. Okay, Shika. So we'll meet in the next class. Have a nice day. Thanks a lot, and let's meet you in the next class.